and good morning. So, different bacteria, and as we've been talking about, have way different growth habits. They're able to metabolize different sugars. Some carry out fermentation in different ways. So lots of different things. And some can be anaerobic and actually operate with different final electron acceptors. Additionally, one of the easy things that bacteria can pass around is antibiotic resistance genes. But this is done primarily through plasmids. And so it's one of the terms we use in the biz of what's known as horizontal gene transfer, which is another one of these things that is kind of foreign because humans really don't carry out horizontal gene transfer. There is some slightly wacko doctor online who's talking about the complexities of viruses in the environment and our interaction with them. And he made some comment about how RNAs are being dispersed by individuals and then taken up by others. And I'm thinking, man, there really is no evidence for that. That's just bullshit. Excuse me. But so we don't engage much in horizontal gene transfer. The injection of an RNA notwithstanding, it just doesn't happen. So we engage in vertical gene transfer. That is old-fashioned reproduction, the way animals do it. Now, bacteria reproduce in a binary fission process, which is their version of horizontal gene transfer, but there's not a lot of genetic exchange. So, you don't have a male bacterium and a female bacterium. The closest we can come with this is this idea of what's called conjugation, which is literally a cell-to-cell actual cytoplasmic connection being made, and DNA can actually be transferred from one bacterium to another. And it only occurs under very specific considerations. Partly, there is a plasmid that is generically known as the F plasmid. And this is required for donors and things. And so if you don't have the appropriate plasmid, you can't generate the kind of sex pilus and the cytoplasmic connection. And so you can transfer this ability to different organisms, and different bacteria can actually participate in this, but not all can. So it is a way that we can transfer DNA. But what we've been talking about here is the sort of basic mechanisms by which we transfer DNA, and they are transformation, which is taking a naked plasmid and bringing it into a bacterial cell. And I don't know if we're going to actually do that in lab, but I do it upstairs all the time and did it this weekend. Then we've got transduction, which is the use of a virus that's able to transmit DNA. And then we have conjugation. And so conjugation does require this specific kind of gene present on the F factor. And that allows the ability of bacteria to actually carry out conjugation. And so you have the F factor can be transferred. You also then have some bacteria have the F factor integrated into the chromosome, and so the chromosomal DNA can be transferred. And one of the things that is done is that you can begin this conjugation, let it go. You have a flask that is carrying out conjugation, and you take samples and you put them in a blender and you whiz the whole thing up, which essentially breaks the chromosomal bridge, and this breaks the cytoplasmic bridge. And it's one of the ways that you can begin actually monitoring the transfer of DNA by looking at what genes get transferred, because they always start at this one specific spot, at the F factor integration spot, and over time, and it takes a while for the entire genome to be transferred over. And you can actually order the genes by looking at what genes get transferred over a period of time. Like every 5 minutes, you take a little sample of the cells, whiz them up in the blender, and you look at the individual time points to see how far did the chromosomal exchange go. And it's one of the ways you can actually begin to map bacterial chromosomes. But it's not something that animal cells do at all. And so it's not a standard thing because it does require the presence of the F factor, which is essentially the gene set for building the cytoplasmic bridge. So thinking about this then, one of the sort of overall questions that we've got from this is how can drug resistance move from one bacterium to another? And so we've got transformation, transduction, and conjugation. And this happens now. It's one of the things that we see, and sort of we presented that antibiotics don't cause mutation. They don't select for, or they don't generate antibiotic resistance. Antibiotic resistance is in the population. It is the application of the antibiotic that begins to select for those guys that are resistant. And once you begin doing this, that genetic mechanism that allows for antibiotic resistance can be spread to a bunch of other bacteria. And so we can even have cross-species stuff happening where you've got bacteria that have been broken apart, their plasmids are floating around, they get taken up by other bacteria, by a complete different species, and now you've got the methicillin resistance gene being transferred to a bunch of different bacteria. And this happens, and that's one of the things that you hear about antibiotic resistance, is that even though one bacterial species may be antibiotic resistant, that gene might be transferred to another bacterium, to another species, and you now have a group of bacteria that are antibiotic resistant. And one of the points I think is often made is that antibiotic resistance is not a matter of if it will happen, but really when will it happen. And so, the common antibiotics like ampicillin, penicillin, are not particularly effective against a lot of bacteria these days. And if you've got a recalcitrant infection, the microbiology expert infectious disease guy at the hospital will begin stepwise cranking up the antibiotic types. And when you get into biz, there are a couple of antibiotics that are pretty much the top tier. You've heard perhaps of methicillin-resistant Staph aureus, which is known as MRSA. And this is a staph bacterium that we all have. We're pretty much covered with staph. And there is methicillin resistance. Methicillin had been a really good antibiotic for inhibiting these infections. It doesn't work anymore, at least not on all staph. And you then have another level of antibiotic. Carbapenem is one of the tippy top antibiotics in terms of not used very often because there are not a lot of resistant strains out there. And the more you use it, the more of the selection you begin to pull out. And so it's kind of a race as the new antibiotics may be introduced. Bacteria are then going to begin developing resistance to it. And so pretty much every antibiotic that comes on the market is going to be generating its own set of antibiotic-resistant bacteria. And, as we said, that you get to a spot where you're practicing medicine the way we did back in the Civil War, because if you can't fight the bacterial infection with antibiotics, about the only choice you got is to actually lop the limb off and hope you can stem the infection from moving forward. Not a good thing. So the different mechanisms are all used. And so here we are. And we can generate resistance by mutation. And these mutations are randomly occurring all the time. And so within the population of bacteria, you're going to find somebody that is resistant to this antibiotic. And that guy may not be a particularly a strong survivor. And so their percentage of the population may be kind of low. But once you start adding the antibiotic, they're the ones that can make it, and they begin to shift the whole population. So that's the way we think about, or that's why genetics is an important part of thinking about the antibiotic mechanism. All right. What we're going to do now, and I'm going to start posting these guys onto Moodle, these PowerPoints. We're going to start kind of a march through the systems and begin looking at some of the different kinds of organ systems and entry points into the human body and how specific bacteria get in there. Because we've got some pretty good bacterial defenses, but there are these openings that allow for bacteria to invade. And we're going to start with respiratory infections. This is a classic spot where bacteria can begin to get a foothold. One of the things I think it's interesting to think about is that really you're covered in bacteria, and if you think of the human body as essentially a donut, we are a toroid, where you got the outside surface, and then with the donut, the hole in the middle, the outside is contiguous with that inner hole, right? And if you think about that inner hole, that's the inner hole. So we also then have these little bags that aren't a tube, but they're infoldings of this outside world, and that's your lungs, your ears, eyeballs. Anything that is a bit of a pocket is a spot where you can get bacterial infection. And certainly the oral to anal gastrointestinal tract, which unfortunately is also hooked up into the lungs as this little sac side piece, that is effectively the outside. I mean, so I think one of the things to keep in mind is that if you were to take a needle and jab into somebody's abdomen, into the abdominal cavity, and pull out fluid or shoot a little bit of saline in and rinse it out, for a healthy individual, pretty much you're sterile inside. There are not going to be cells that aren't you inside you. I mean, you can have cancer cells that are moving around, but they began as you. And so the same thing with blood. If you draw blood, venous or arterial, and there's bacteria in it, that's not good. And for most, for healthy people, there is nothing inside there except you. I mean, there's some interesting things about after pregnancy, mothers can actually have fetal cells floating around in them. But in terms of bacterial pathogens, fungi for that matter as well, inside it is all sacred stuff. So every time you break open a donut, just realize that part, the dough part, that was separate from the outside world. That is what the internal systems are. And so we can begin looking at these different either tubes or pockets, sacs, and see what kind of infections can occur there. For that matter, we'll be looking at the surface as well. There's all kinds of skin surface bacteria as well. So we can think about this from a couple of perspectives, is that you are covered in bacteria and it's necessary for health. You can strip the bacteria from your surface using a variety of harsh chemicals, but it is not safe. It's not very good for your skin. It also means that your skin is now wide open and lots of stuff can get in there. And I kind of think about this as like our bodies are really one great big bacterial parking lot. And you want to make sure that your body is filled, all the parking spaces are filled with happy non-pathogenic bacteria. If you've got open parking spots, you might have a methicillin-resistant Staph aureus coming in, or different kinds of bacteria that are not very helpful. Additionally, and one of the things we're going to see is that one of the things that can happen if your immune system gets a little bit slow in kind of cleaning things up, the bacteria that are typically always around you get a bit more of a foothold than they really should. And so if you were to do a nasal swab or a throat swab, you would find that we all have pneumococcal bacteria in our respiratory system. And so this is a bacteria that causes pneumonia. And it's not unexpected to have that. I mean, it is part of the natural bacteria surrounding us. The problem comes when you have sufficient immune compromising issues, the flu, whatever, stress, and those bacteria numbers start to get to be too high. And that's when you have a problem. So most of the time, at least with a bunch of infections, it's not that you have picked up something new. It is that you have allowed your immune system to not back everything, keep everything in check. So as we know, the bulk of bacteria are either harmless or useful. It's one of the things that people are encouraged to eat yogurt, and If you've been on antibiotics, you're encouraged to eat yogurt. Yogurt is filled with a variety of non-pathogenic, useful, cover yourself in bacteria-type bacteria. And one of the things I often wonder is, like, when you eat yogurt, it is a massive bacterial mix there. And it's possible that the act of opening it up creates enough aerosol, and eating it excuse me, begins to spread it around you, that you're actually providing yourself with this surface type of bacterial infection going on. And so there is, pretty good reason to think about yogurt as not only a useful food, but actually giving yourself a really good bacterial infection. Just, as an example, that when you've been on antibiotics, or if you've had, like, for women with a yeast infection, you're encouraged to eat yogurt. And I'm always going, is it that, how is it that the internal consumption of this bacteria is getting around to different pockets, shall we say? And it's probably very likely that when you crack that thing open, there is sufficient bacteria floating around from that you're giving yourself a pretty good, reinfection. So I guess if you don't like yogurt, stand next to somebody that is eating yogurt. Or crack open a container of yogurt and smear it around on you, and then, so you don't have to eat it, just be really near it. So the bacteria that live on us actually are pretty tough guys. They have to maintain, they have to be able to survive all these different kinds of defenses and things. And so we've got then some disease issues. And so we can really, I think about it in a couple of different ways. There are the bacteria that have virulence factors that are going to make you sick. And that just sucks. Then there are bacteria that are on you, and they start to get a little bit too much on you. They get a foothold in the body that is higher than what the body can handle. And that's how you can get things like pneumonia. So what we're going to start doing is working our way through some standard bacterial types and how they're causing infection. And this idea is that we have bacteria that are around us all the time. And it is more or less a symbiotic relationship in that the bacteria have a place to live, they are able to walk around and be spread about, and in turn, they either are not bothering you, or they are providing all kinds of beneficial properties, like the production of vitamin K requires bacteria in the intestines. So it just isn't possible to generate a person that is devoid of bacteria. You can try it, but it would be a really, you probably can't survive very well without having these bacteria on you, which is kind of interesting to think about. You can try and make yourself as sterile as possible, but there's a limit to how much you can do without actually making yourself sick. So when we think about symbiosis from a biological perspective, we have this idea of mutualism, where both members of the relationship benefit. Humans and dogs, sure. But then we've got other kinds of symbiotic relationships, like commensalism, where one guy gets a benefit and the other guy just isn't bothered at all. So, I don't know, humans and seagulls? The seagulls are generating a tremendous gain, but they don't bother us. You feed them. The cattle egret and the buffalo, they are living together, and the buffalo doesn't really mind much. Doesn't really get a whole lot out of it, but the bird is picking up after it. Barnacles hang out on whales. Yeah. So the third kind is not necessarily the most happy kind of symbiosis, but it is a type. And this is parasitism. And this is where you got one member of the symbiosis benefits, and the other individual isn't just left alone, they actually are harmed. And this is a delicate balance. So we've got, like, the bacteria that can cause cholera can hang out on you and actually survive in you and replicate in you, but it gives you a real serious disease. Tapeworms in humans and dogs. Mosquitoes are another example of parasitism, where they are They require the blood meal in order to actually be able to lay eggs, and they have the ability to transfer diseases and things. Parasitic wasps are all around and interact with lots of different insects. In this case, there are certain wasps that actually will parasitize agronomic important pests, and so uses a bioremediation system. So it depends on which side of the equation you're on, whether this is a useful thing or not. So one of the games with parasitism is if the parasite is too aggressive and kills the host too fast, it's not very good for the parasite because they don't have the host anymore. So you have to have this delicate balance of, I'm getting something out of you, you're not very happy, but you're not so unhappy that you end up dying. Or hopefully you're not so unhappy that I cause sufficient difficulty that you get rid of me. So a lot of parasitic interactions can be a little bit quiet. They have long-term effects, but you may not notice it immediately. So then, another kind of definition we can think about that we need to apply to our whole way of thinking about disease-causing bacteria and these different systems is the progress. So it does change for sure, depending on what organ system and things you're talking about, and what kind of bacteria you're talking about. But we have a similar set of steps in this progression. And so incubation is where we begin. And this is the initial spots where you don't have symptoms, but the pathogen is in you and is starting to replicate and get enough, beginning to take advantage of the situation. So the pathogen begins to replicate, damage starts to happen, and the prodromal stage is referred to as the first stage in which you get the inkling that, I have some kind of issue here. The quote, generic flu-like symptoms. One of the things that we know, you can demonstrate this for yourself with vaccines, is that the vaccine itself is not, in the case of the COVID deal we're working with right now, the RNA virus, or the RNA piece that is in Moderna and Pfizer, is not able to replicate and is not able to act as a replicating infectious virus. And it doesn't even do the same things to the cell that the virus does. What you're experiencing when you have the various sorts of aching and low-grade fever from the vaccine is exactly this kind of not reaction, or not a consequence of the organism, but a consequence of your reaction to the organism. And so these kinds of flu-like symptoms, body aches and pains, being tired, these are symptomatic of any kind of immune response. So it's what we would expect at the early stages of some kind of disease progression. So then we get to illness. Now this is where we have differences in the organism that's causing the disease. It's no longer the immune response. You've now progressed to what is the disease doing. And so in the case of COVID, because it infects the lungs, we begin to have respiratory issues. The initial infection is going to generate this sort of flu-like symptoms, but you can mimic that with a vaccine. What's next is something that the vaccine doesn't do, and that starts to have all kinds of respiratory infection problems. So we have then the symptoms that are characteristic of the disease. Could be diarrhea, could be boils and pustules, who knows what. So the immune system's job at this point is to try and knock back the pathogen. If it doesn't, that's when you start to have more serious problems, death being one of them. All right, so then if the immune system is able to get a hold of things and start taking back control, you have the decline. The symptoms begin to lessen, and you start feeling better. Convalescence is the stage that we characterize where the symptoms are gone, but you're still pretty wiped out. One of the things that happens is that the act of going through this is challenging your immune system, and it causes a weakened immune system, and it opens you up to secondary infections. And so a standard thing that often happens with the flu is people will get the flu and as a viral infection, as we know, pretty much the only way you knock back a virus is through a robust immune system. If your immune system has got a handle on it, you're going to get better. If you're immune compromised, you're going to be in serious problems. The fact that your immune system is having to work and being taxed, it opens you up to secondary infections. And so people then will get pneumonia, after the flu. And there is viral pneumonia, which you can actually catch, but then there is more common bacterial pneumonia, and anything you catch, you already had it, the pneumococcal bacteria in your lungs. It is simply that the immune system has been weakened, and now it takes off. So it's one of the things that, maintaining your immune system is probably one of the best things you can do for your health. How do you maintain your immune system? Try to reduce your stress, sleep more, eat well, all the things that we probably tend to not do very well. And, for viruses, that's pretty much your only defense other than a vaccine. So maintaining a good immune system is important. Prior to the age of antibiotics, you either had your immune system taking care of it, or you had, the removal of a limb. So it was your choice. All right. So again, a general way of thinking about bacterial or infections in general is these steps. You have to have some portal of entry. You have to be able to evade the defense system, somehow not get taken out by the body's defense system. You have to be able to grow in us, and that's not always that easy as well. You can interfere with our metabolic processes as well, and that results in the symptoms. Another one of the goals of bacterial infection is to reproduce sufficiently so you can be spread, and then exit the bodies and make their way to the next host. So this is the kind of mechanisms that we'll be looking at of how these bacteria or these various disease-causing agents get in, how they're evading, and then particularly how they exit and how they are able to be spread. So if we break apart these portals of entry, they are critical because where a pathogen gets in is going to be a critical part of what's going to be happening, of the symptoms. And just as an example, you got the nose and mouth. This is the entry into respiratory diseases. The mouth also is an entry spot for digestive diseases. Skin, your skin, as long as it's maintained intact, is a pretty good repellent. But tears, little cuts, different kinds of bites are portals for entry of disease. The urogenital tract. We can have a variety of different kinds of infections that can get up inside you or by different routes and things, but end up in the reproductive tract and the urinary tract. There then are these other little pockets, the ear, the eye, the anus, the follicles of your hair, are other spots for portals of entry. And these are all spots where bacteria and different infectious agents can get in. So, you're not defenseless, because initially this feels daunting. We have the exterior surfaces. We have our skin. We've got different kinds of behaviors. There's movement of different kinds of cilia in your lungs are constantly knocking things up, causing you to cough and expel dust and various sorts of bacterial bits that may be on the dust. Internally, we've got all kinds of different secretions and things. We've got a whole range of different movements and such. So it's one of, again, the thing is that if you have a very low urine output, if you're not peeing much, you're at risk of a urinary infection because you're not flushing stuff out as well as you need to be. Not being able to move food appropriately through the intestine and have a poop is another problem that you can end up with. So there's a lot of different kinds of mechanisms that we use to keep the different kinds of bacteria and things from taking over us. So deep inside, within the donut, we've got enzymes. There are a whole host of antibacterial proteins that are found in body fluids. And then we've got different kinds of immune response systems. And we'll be talking a little bit about that actually, not if time, but for sure, talking about the immune system because it's a very cool topic to discuss. So, all right. We got then a number of different mechanisms by which we can have these microorganisms, bacteria, causing disease. And again, some definitions of virulence is the ability to cause a disease. How virulent is a pathogen? And then what makes it virulent? These are the virulence factors. And so these are the kinds of things that a bacterium, virus might have that give it the ability to cause a disease. So you can have, this is an example, the old-fashioned vaccine for polio. There was two different versions of it. There was the heat-killed form, which they just autoclave it, but then there was an attenuated form in which they had passaged the virus sufficient times to begin picking up mutations and generate a virus that is not particularly virulent. Now, when you inject that, your body is able to very easily overcome it, and it serves as a really good vaccine. And so there are some pathogens that are really, really virulent. And one of the thoughts is about, in terms of the arc of the COVID infection, is that as a coronavirus, there are a number, at least half dozen or a dozen or so, viruses that cause the common cold that are in fact coronaviruses. And probably over the past, couple thousand years, these kinds of coronaviruses have entered into the population, been really devastating, and you can imagine that if you didn't have adequate health care, we'd have much more serious problems. But regardless, the virus is in the population, and then over time, its virulence begins to decrease, and it becomes nothing more than a common cold. And so that's one of the ideas about, or the predictions about the coronavirus that we're currently struggling with, is that in the next 5 to 10 years, this virus is probably going to mutate in a way to attenuate its virulence. And it's going to become less debilitating and more like a common cold. So what we're seeing is the introduction of a virus that eventually will give you the sniffles. So that's takes some time, but that is one of the most probable outcomes for what's going to happen with this virus. So some of the different virulence factors are worth knowing just because they determine how we can begin to have some kind of therapy. So the offensive virulence factors, these are the things that are really dangerous. That's what's going to make you sick. Things like endotoxin. There is a component of the bacterial lipid makeup known as lipopolysaccharide. Is just the general term that's found in gram-negative bacteria, LPS. And lipopolysaccharide is a pretty nasty substance to inject into somebody. There are exotoxins. Usually they're gram-positive. Gram-positives just have a more elaborate candy coating. And as we were looking at the Avery Griffith experiment, where they were looking at the smoothened versus rough bacteria and the ability to actually kill the mice, this is an example of an exotoxin, in that the smooth bacteria had the exotoxin present in the candy coating. The rough guys were missing that. And so just having that actually can generate some different kinds of diseases. Again, you can have, trying to think of what it is. MRSA can cause a Staph aureus can do this, where you have a boil that begins to sort of spread, and you can end up with a region of the skin that is becoming necrotic as the bacteria secrete these digestive enzymes, and they essentially begin dissolving you from the outside. Proteases, nucleases, these are things that begin to degrade the tissue. Viruses are particularly good at lysing cells. So we can have cells essentially being exploded by the virus. That doesn't go very well for anybody. We're not defenseless, though. We have the other side is different kinds of, well, actually, they're not defenseless. Other ways of sort of acting stealthily to escape the immune response. And so bacteria often have capsules and proteins that will allow them to evade the immune response. There are materials on the bacterial surface that allow them to stick really tightly to like the inside of the urethra so they don't get peed out. You can have bacteria and viruses and actually some parasites, some animal, eukaryotic parasites, that have what is known as an antigenic shift, where they're presenting a protein on the cell surface that the immune response can actually begin triggering off of, and then really in a matter of a couple of hours, they can shrug that stuff off and change it. And now all of a sudden, the immune response no longer works, and they are essentially changing sort of, quick change of disguises and stuff. There are also pathogens that are specific at degrading antibodies. Not a very good thing. There are also a whole range of virulence factors that are allowing a virus to get into different kinds of cell types and things. So the other side of this is getting out. We have the getting in mechanism, but it doesn't do you much good as a pathogen unless you can get out and spread. I mean, it's one of the things that you have to be thoughtful of, is it's easy to anthropomorphize or to place human characteristics on these things, and that's just not how the system is actually working, but it's a convenient way to think about it. And so the exit strategies, you provoke different kinds of symptoms like sneezing, coughing, spitting, runny nose, diarrhea. Oh boy. So this is a way you can get the pathogen back out into the environment. That's why we're wearing masks. You can have the bacteria hang out at places where materials are released from the body normally. So things like, being in the intestines, so you're not causing diarrhea, but you just hang out with the poop. The bladder is a great place to hang out and spread. It's not necessarily causing you to pee, but they go ahead and hit you right there. Different digestive parts in the mouth. Lots of different ways you can do that. Another thing you can do is to essentially encyst in the tissue. And is wait. You can have the gastrointestinal tract as another spot where you can just hang out. And this is things like primarily like tapeworms and stuff. You can also hang out in a whole range of different body fluids and just wait for them to be released. Another way of spreading is you have a vector like a mosquito that gets into your blood, pulls blood that has malaria in it, and then transmits it to the next host, whoever he happens to chew on next. All right, so we will continue on these and see where we go from here, but we'll stop here.